Am I audible? Is the voice clear? Good evening, Imran. Hello, Jay. Okay. So I'm just using my phone screen for looking at your messages. Okay. So whatever you type, I'll see here. Okay, and I'll be replying because I cannot go back and check on the laptop. So we'll be starting the discussion. I think in just 2 or 3 minutes. Okay. It's almost 6 o'clock here. Okay. So am I audible? Fine. Is the voice clear? Okay. Anamika, Rupesh. Hello. Fine. Okay. So we'll be starting, okay, and as you all can remember, okay, those who are in class, okay, you attended the class recently, okay, almost all question you could have solved using the knowledge or information available in the notes, okay. Some question like the question about hemoglobin is question related to also the concepts of biochemistry, okay. So that's why there is always a amalgamation, okay. You can never know this question is exactly from physiology, okay. So that's why there is always a mixture of all these things that is possible, okay. So we'll be discussing the questions. Okay, fine. Okay. So once again, hello to all of you. Okay. So I'll be starting the discussion. Okay. So let's start. Okay. So they were around 11, some said 13. So we have taken all questions. Okay. Even some were a part of both psychiatry also physiology, because I told you there are things which are always overlapping. Okay. So we'll be starting. Okay, so the first question I'll discuss is something which I already informed and gave you in the beginning itself. Which of the following hormones have intracellular receptors? Okay, and something which I discuss always in class. Okay, and I also, so in fact, if you remember in the class itself, I will do this. Okay, and if you can see, okay, this was the mnemonic, okay, and it used to help you solve another question regarding the priming also, okay. So you can remember this photo about the dirty pig, okay. You might be able to remember what I discussed there itself, okay. So dirty pig was a mnemonic. So let me first explain this in a simplified manner, okay. So as I told you that any hormone that we have, okay, so the hormone would be having basically two types of, you can say, things here, okay. So what are the two things that we see? Okay, 1 second. Okay, I just need to start my tablet. Okay, 1 second. Okay, yeah, fine. Sorry for that. Okay, just a second. Okay, let me just connect it with cable. So now it is fine. Okay, sorry for the delays. Okay, it's just okay. So I hope now am I audible, visible? Okay. Okay. So sorry for the interruption. Okay. Sometimes things don't go as planned. Okay. So my pen was not working. Okay. So as I have discussed already in the class many a times. Okay, that we have two kind of hormones. Okay, so they are basically what? They are lipid soluble hormones and water soluble hormones. Are you clear? So they are lipid soluble and water soluble hormones. Okay, now what is the difference between the two types? Okay, first is the lipid soluble are basically thyroid and steroid. Are you clear? So thyroid and steroid are the lipid soluble ones, and the water soluble are basically what? They are peptides, polypeptides, or we can say peptides. So polypeptides. Are you clear? Polypeptides, pp. And I told you pp, pani is water soluble, so pp is water soluble. Are you clear now? Okay, and I also gave a hint that mostly they end with, mostly not always, mostly and with in H or P. Yes or no? So discuss that they mostly end with in H or P. So that is the idea in this case. So they are what? In H or P. Okay. So in H or P is the term here. Okay. So in H and P. Okay. Clear? So in H and P. Okay. So insulin, ghrelin, vasopressin, oxytocin. Okay. Somatostatin, prolactin, all are polypeptide. Then H1, LH, FSH, TSH, GnRH, CRH, PTH, or P1, GLP, GIP, NP. So all these in H and P are primarily polypeptides. Are you clear now? Clear? Fine. Okay. Even what monoamines act through this, even monoamines, because they are also water soluble like epinephrine, norepinephrine, serotonin. So they are all going to act. So thyroid, steroid at what? They are lipid soluble. They are lipid soluble, and they are going to act through this. Okay, now first is they can cross cell membrane because they are lipid soluble, cross cell membrane, and therefore they act directly inside cell. Are you clear? So they are going to act directly inside the cell. Okay, so they will act directly inside the cell. Okay, whereas polypeptide can't cross cell membrane, can't cross cell membrane. So that is why what they do is they basically use cell surface receptor. So that is why they use cell surface receptor. Are you clear? So they use the cell surface receptor. Are you clear? Okay. So they can't cross the cell membrane and they use what? The cell surface receptors. Okay. Clear? So cell surface receptor. Okay. Which one? These ones. Okay. Now here comes the question, what is the concept about the cell surface receptors is. Okay, so first is for the cell surface receptor. Okay, let me explain. Okay, so they are basically two types. Okay, so cell surface receptors. Okay, so question is this one. Okay, so which hormones use intracellular receptor we are discussing. Okay, so summary is, okay, so thyroid, steroid, they act directly inside the cell. So act directly inside cell. Why? Because they are lipid soluble. So they are which group? They are lipid soluble. So they act directly inside the cell. Are you clear now? So they are acting directly inside the cell. Okay. So let's come back again. Okay. So act directly inside the cell via receptors. Okay. So which receptors? Let me clear that. Okay. So basically thyroid, vitamin A, D, they are nuclear receptors. Now pharma books or some authors, they say that vitamin D act through cytoplasmic receptors. Wrong. Okay, vitamin D act through steroid superfamily of nuclear receptors. So vitamin D is nuclear receptor. Okay, whereas the steroids, okay, the sex steroids, okay, and glucocorticoids, whereas the mineralocorticoid, aldosterone. So aldosterone is only cytoplasmic receptor. Are you clear? So only and only cytoplasmic receptor is the case. Are you clear? So only cytoplasmic receptor. Okay. Fine. So they are going to use only the cytoplasmic receptor. Okay. Aldosterone and this one. Okay. So is the summary clear? So summary is which hormones are they going to use or they'll be having. This is the basic idea in this case. Okay. So glucocorticoids. Okay. Aldosterone basically uses what? Cytoplasmic receptor. Okay. Done. Okay. Then I'll make this slightly smaller because it's coming in the middle. Okay. So that is the idea here. Okay. Now what they act through. So sex steroid and gluco act through both. So they act by both cytoplasmic and nuclear receptors. Are you clear? So they are acting through both cytoplasmic and the nuclear receptors. Are you clear? So both cytoplasmic and nuclear receptors are used in this particular case. Okay. So cytoplasmic and nuclear receptors. Okay. So this thing is done. So that means thyroxine T4 is going to act through what? Nuclear receptor. So the answer for this question is nuclear receptor. So it is N. I'm writing N. Okay. Now let's see the remaining hormones. Okay. So as I told you, other hormones which are not thyroid and steroid, they are basically the water soluble ones. So water soluble are basically polypeptides which are ending with in H or P. So PTH is a polypeptide, is ending with what? With H. So it is polypeptide. Okay. Insulin, they all are. Okay. So what do they act through? So let me just clarify it. Okay. Okay. So remember, cell surface receptor basically has two types. One is the tyrosine kinase couple and other is GPCR. Okay. There is G protein coupled receptor. G protein coupled receptor. Are you clear? So G protein coupled receptor is the term. Okay. So G protein coupled receptor. Okay. So G protein coupled receptor. Okay. Now question is who use what? So remember tyrosine kinase is used primarily by three hormones, that is PIG. Okay. So three main hormones. So PIG, that is prolactin, insulin, and GH is growth hormone. Are you clear? So prolactin, insulin, and growth hormone. Okay. So they are polypeptides. See in and H. So they are polypeptide in H. Okay. And GPCR and GPCR. Okay. So GPCR I'll discuss in just some time, but the remaining are all GPCR. Okay. So you can say they are mainly non-pig hormones. So non-pig. If you want to remember, non-pig. Okay. And if you remember in the class, I always used to give this mnemonic that remember that mnemonic is pig. Okay, clear? So pig. Okay, and even I used to make some diagram of the pig. Okay, remember? So this pig I used to make. Again, it's an example of memory formation. Okay, so this is the pig. Okay, and we know pig is what kind of animal? Dirty animal. So what is the purpose of TY? That TY basically indicate what? Tie. Tie for tyrosine. So you know all pig is a dirty animal. So pig, dirty, tie. Tyrosine is pig. So that was a mnemonic we learned in the class. That's dirty pig. Okay, clear? And then I said in the class when I was, and I told you that this is something I also shown in the class itself. Okay, see. So I shown in the class the dirty pig is going to 100%. See, I even wrote 100% there. It's a 100% question in the exam, and it was there in the exam. So 100% question is there from the topic of pig. Okay, so dirty pig. Okay, and just added some extra points. What are the extra points? Let me just say. So extra point which we are having is, okay, plus GF. Plus GF. What is GF? Growth factors. Growth factors. Okay, so you can just recall if you want to make that dirty pig and dirty pig with the GF. Okay, so dirty pig and dirty pig GF, you can memorize or imagine a GF sitting on the pig something. Okay, not with the pig. Okay, sorry, not with the, okay, so GF is growth factors. Are you clear? So GF is growth factors in this particular case. Are you clear? So GF is growth factor in this case. Okay, so what growth factor? Example, IGF, NGF. Then we have what? Yes, the this which one? The epithelium drive growth factors, fibroblast growth factors, okay, etc. So all these again use the tyrosine kinase. Okay, clear? Fine. Now some people keep asking, sir, but we read something extra also. Now remember, insulin is a metabolic hormone, so its pathway of action is slightly different. So if you want to remember, I can just add one more information, and that information is, if you write it here. Okay, that, okay, that prolactin and growth hormone, they basically, tyrosine kinase, they use JAK-STAT pathway. JAK-STAT pathway. Are you clear? And increase the protein synthesis. Are you clear? So they use the JAK-STAT pathway and increase the protein synthesis. Are you clear now? Increases the protein synthesis. Okay, clear? Fine. So what happens to proteins? So proteins are going to be increased. Are you clear? So proteins are going to be increased in this particular case. Okay, done? Okay. So is this thing clear? Okay, so proteins are going to be increased in this particular case. Okay, so now for clear, so prolactin and growth hormone, they are using JAK-STAT pathway. Are you clear now? In fact, growth factors will also be using JAK-STAT pathway. Okay, insulin is using IRS, that is insulin receptor substrate. So insulin use IRS for its action. So insulin, it uses IRS. So now it's summary of everything. Okay, it use IRS, insulin receptor substrate. So I think everything is clear. So insulin use IRS and rest all use JAK-STAT. Okay, prolactin and GH. Okay. Now GPCR would be all. So which one? Any hormone which is outside is this, like LH, FSH. It will be which category? It will be this. So example, LH, FSH, TSH, MSH, ACTH. Okay. So all, okay, GnRH, okay, FRS. So this will become a very long category which will, yes, we will see here. Okay, so they all would be there in this group. Are you clear? So all LH, FSH will belong to which category? GPCR. Are you clear? GPCR, and it's a cell surface receptor. Okay, clear? Okay, and if you remember, I have given even the summary of GPCR also. So in case of GPCR, you have to remember there are three categories. So what are the three categories of the GPCR? There are three categories, which are, they are basically cyclic GMP, then cyclic AMP, and IP3DAG. Are you clear? So these are the example. Okay, GMP, AMP, and IP3. Okay, and what are the examples as such? So remember this thing here. GMP is NO and ANP. So nitric oxide and this, okay. Cyclic AMP, I'll leave it for now because it's something, and this basically contraction hormones, contraction. So this cause basically contraction. So this cause basically contractions. Which one? Oxytocin, yes or no? Vasopressin, angiotensin, endothelins, then gastrin, cholecystokinin, so contraction. Even acetylcholine, okay. So they are causing contraction. They use IP3 and DHE, okay, clear? So I don't have space to write. And cyclic AMP is basically the remaining, okay, or the rest, okay, rest others. So given it very clearly in the class, I took almost half an hour to discuss all the receptors and their actions, okay. So right now we cannot spend half an hour for this. It's just a summary, okay. So therefore NO, ANP, cyclic GMP, okay. Cyclic AMP include all others. So LH, FSH, TSH, ACTH, they will all belong to which category? Cyclic. Glucagon, cyclic AMP, okay. PTH, cyclic AMP. Calcitonin, cyclic AMP. IP3, contraction hormones are the ones here, okay, clear? So even epinephrine, norepinephrine alpha 1 cause vasoconstriction, okay. Acetylcholine M1 and M3, M1, M3, again it uses this particular things. Are you clear? So it will be using this. Is it clear now? So it is the idea here, okay. So everything is crystal clear, okay. Even which ones? Contraction, oxytocin, angiotensin 1 and 2, mainly 2, okay. Then our yes, vasopressin, ADH, which cause vasoconstriction, and also, so that's why all these hormones belong to the category of IP3 DHE, okay. So let's see the answer here. So as we have seen, glucagon would be acting through what? It acts through cyclic AMP. Epinephrine, it used through both IP3 also, IP3 DHE, which one? The alpha receptor, okay, and cyclic AMP, the beta receptor. Are you clear? So it is said that it uses both beta receptor use cyclic AMP and alpha receptor use IP3 DHE. Parathyroid regulate the calcium, so it's a regulatory hormone, so it use what? Again cyclic AMP. So these three are what? Cell surface receptor, rather better word GPCR. So the answer is yes, A thyroxine is the answer here, okay. So A thyroxine, okay. So let's move, okay. Next is in a negative feedback gain is infinity in which of the following, okay. Now this question requires some of very important basic information. So I'm going to say, and please remember, first is misconception number 1. Negative feedback means decreasing. No, negative feedback means going away from the change. Are you clear now? So first understand, okay, negative feedback means, okay, going in opposite direction to the change, okay. So first it is opposite direction, okay. Like example, if BP is increasing, baroreceptor decrease the BP that is an example of negative. But if the BP fall and now the barochemo try to increase, that is also example of negative feedback. So example, increase BP, then the baroreceptor reflex, baro will decrease BP to normal. This is also negative feedback. And if BP is falling, decrease BP, and now again baro kidney, RAS, and they increase the BP by conservation of fluid. Again, it's an example of what? Negative feedback system. Are you clear? So it's again an example of negative feedback system. Okay, clear? So they both would be included in which category? Negative feedback system. Are you clear? So negative feedback system is the case. Okay. So first, negative feedback is always what direction? It is the reverse direction in this case. Are you clear? Okay. So it is a reverse direction. Okay. Okay. So I hope it's clear. Okay. So it is the reverse direction. Okay. Now next question. And remember, AIIMS question, if you see the previous question, you'll get an idea about the future questions. Okay. So there was a question about the anticipatory control last year number exam. Okay. Temperature regulation is example of again negative feedback, but it's feed forward, not feedback. Okay, so feed forward regulation. So now question is, first let me explain some of the important concepts. Okay, what do you mean by the concept of gain? Okay, so let me explain it. Okay, so what is gain? So first understand, for testing efficiency of any regulatory system, we use gain. We use the concept of gain. Are you clear? So the gain is the concept we use for this particular phenomenon. Are you clear? So we use the concept of gain. Okay, so for testing the efficiency we use the concept of gain. Okay, now what is gain given by? So gain is given by correction done and the remaining error. Okay, error which is still remaining. Okay, okay, not the error in the beginning, because if that is the case you will have something else. Okay, so correction by error. Okay, so correction by error. Are you clear now? Okay, now question is okay, first let's just take an example is, let's say BP normal is 120. So what the normal will be 120. Okay, is the normal BP. Okay, is the BP. Okay, mmHg systolic BP. Okay, now because of, let's say, exercise or something, it rises to, let's say, 170. So what is the BP rising to 170? Okay, so it is the error. Okay, total error which started. Okay, so it become 1. Okay, now baroreceptor correct it back. So correct it back to, so it will correct it back to, let's say, 100 and let's say 30. Okay, so what happens? So they correct back to 130. So now if you pay attention, what is the correction they have done? So what is the correction? So correction is 40. So that means they did a 40 mmHg correction. Okay, so 40. And what is the error still remaining? The BP was 120, so error still remaining is 10. Okay, 10. So the gain will be 4. Are you clear? So that is what the idea of the gain is. Okay, clear? So gain is this. Okay, friend. So 40 you reduce the pressure, 10 is still the, this it's an example of negative because the value would be minus. Okay, so it will be a negative. Why? Because you are decreasing it. So you can just say by checking the value it's negative or 40, you can know it is doing which change. Okay, so I'll just remove it right now because not required. So first is we are doing a gain of 4 in this case. Okay, now question is, if the BP become 120, okay, if the BP become 120, that means there is a correction of 50. Are you clear? So correction of 50 and the error is 0. And the error is 0. And we know from mathematics that any number divided by 0, again it was given first time by Indian scientists. Okay, clear? I think it's Madhvacharya. Okay, clear? So okay, he gave that any number divided by 0 will give infinity. So obviously correction cannot be infinite. You cannot. Why? Because all biological systems have a limit. Okay, like can you become as high as a mountain? No. Why? Because the laws of physics will apply. As your body size increase, your mass will increase, and you know mass and area is the relation of what? Square and cube. So therefore as your mass increase, your body surface area will be much lower than the actual body mass. So therefore your cells will die of hypoxia, plus the gravity will crush all your body tissue and you will be not able to even move an inch. So that is why it's not possible to make a human the size of mountains. Are you clear? Clear. So that is what the idea is. Okay, so a cell has a size, whether it's an elephant, blue whale or human or a mice, the cell size is almost same, some micrometers. Why? Because again laws of physics takes over here. So therefore the correction cannot be infinite, so you cannot have. So therefore to get infinity gain, the best way is make the process so perfect that the error is almost 0. Okay, clear? and that means you have a very big range of error correction. Are you clear? Fine. Okay, now here comes the question. So what system is capable? Okay, technically infinity gain is impossible. Okay, why? Because always some error is left. Okay, the only system which is given in physiological books, especially Guyton, that which has an infinite capacity of correction is our kidneys. Okay, why? Because the kidneys are regulating a huge amounts of blood which is being filtered 180 liters a day. So therefore let me just go through this and then we will learn and understand. Okay, so first understand. Okay, temperature regulation is basically mainly not an example of negative feedback. It's a feed forward. It's a feed forward or anticipatory control or anticipatory control. So what happens here? You correct the temperature even before it actually change. So here what happens? It's a feed forward. So you are not waiting for your body temperature to fall, then you will give the correction. So we correct it even before that. Are you clear now? So it's an example of what? It's an example of feed. Which one? It's an example of which one? Feed. Is okay, fine. Okay. So I hope you are all getting the idea. Okay, so therefore yes, it is feed forward. Okay, in feed forward the again is not possible to calculate. Why? Because the core temperature never change. Are you clear now? So therefore the correction will come 0, and if 0 correction, so therefore what happens? Okay, the correction is given even before the change. Okay, so correction is given even before the change. Are you clear? So correction is given even before the change. Let me give example. Okay, the core body temperature, let's say is 37 point something. Okay, so this is the core body temperature 37. Okay, now if you go to a cold environment, now it doesn't matter how cold it is. Okay, so when you go to a cold climate, what happens? The shivering start, vasoconstriction occur, piloerection, all these are taught in this. So now what happens? They will occur immediately when you enter the cold environment. Okay, and the result is your body start making more heat, and the result is what happens? So this will increase temperature and prevent fall. Prevent, it is not correcting the fall, it is preventing the fall. So it will prevent change in temperature, prevent fall in core temperature. Are you clear? So the core temperature is not going to change. Are you clear? So core temperature is not going to change at all. So what is the correction? It is given before. So therefore what is the change? The error. So that means there will be almost no error or a very small error. Are you clear now? So therefore you cannot have what? In this case the concept of this infinity. Okay, fine. So that is one thing done. Okay. Now comes the question. Question is blood volume control. Okay, so I told you already that kidneys have almost perfect regulation of the volume because they have both. First they can gain the volume and lose. Okay, so they have what? Almost infinite correction and gain. Okay, clear? And what is the system they use? First, they can regulate the volume and therefore they always maintain. Like example, if there's too much volume, like if there is increased volume, okay, there would be increased GFR, increased urine volume, and they will keep making extra urine till the volume is almost normal. So therefore, and normal volume. Okay, so that's why they will do normal volume, and therefore here the error is 0. So again, what error? 0 error. So that make the infinite gain. Are you clear now? So therefore correction is perfect and it is infinite this. Are you clear? So I hope it's clear now. Okay. Now next, is it clear now? Second concept is, and if the volume is too low, it will decrease GFR, also use the RAS, and the result is what? There would be more absorption of water. There is decrease in the, you can say, the more absorption of sodium water, less urine. So now there is what? Decrease urine. okay, decrease urine till there is normal volume. Now what I am trying to say is understand, okay, again this is almost perfect, okay, but if it was a foolproof system you will never have shock. But does shock occur? Yes. Why? Because eventually you cannot correct it completely. So therefore yes, hypothetically it is having a physiological infinite gain, but it is again having limited, okay. So out of the best answer is kidney, because it is having the largest range, okay, almost it's said to be infinite but is not infinite. Are you clear now? Okay. Now BP control, as I have already discussed, it is not having an infinite gain. Why? Because there are other factors also involved and baroreceptors never correct fully, okay. So some error is always left, okay. So some error is always left. So some error is always left. So that's why out of the three, the best answer is B. Okay. Now D, such infinite feedback gain is not possible. Okay. Technically statement is right, but since it is given in books that kidney possesses almost perfect correction, so therefore the best answer in this question becomes our, which one? It is our kidneys. Are you clear? Because we have reference, that is Guyton, which is giving it the case. Are you clear? So Guyton is the answer. Okay. So next is Gibbs-Donan equilibrium is mainly by, okay. So let me explain first, understand what is the concept of the Gibbs-Donan equilibrium, okay. So first for Gibbs-Donan, understand a very important point for that, okay. Gibbs-Donan, okay. It is basically due to what? It is basically due to indiffusible ions. So it is basically due to indiffusible ions, okay. Now who's the main, okay, that I'll discuss in just some time. So basically it is due to the Gibbs-Donan equilibrium. Are you clear now? So Gibbs-Donan equilibrium is the case, okay. Gibbs-Donan, okay. Now question is, what do you mean by that and what are the concept? So first understand there are two kind of ions, diffusible, indiffusible, okay. So first is diffusible ions. What they can do? They can cross cell membrane. They can cross the membrane. That is example potassium and chloride. So therefore they are the ones who are able to move, and that's why they are responsible for RMP. So that's why they produce RMP. So they ask in the exam, who causes the RMP? The answer is RMP is caused by the diffusible ions. Are you clear? So RMP is caused by the diffusible ions. Are you clear? Okay. Whereas the indiffusible ones, okay, they cannot move. So therefore they are the ones responsible for Gibbs-Donan equilibrium, because they cannot move. Diffusible move in such a manner that there is always a equilibrium achieved. So they are which ones? They are basically sodium, calcium, okay, proteins and phosphate. So first thing is, if they ask you is caused by, the answer is indiffusible ions cause the Gibbs-Donan, okay. And what causes the RMP? Diffusible ions, that is potassium and chloride. They are producing the RMP, okay. Now what is the equilibrium? So understand that total number of cation and anions is equal. This is equilibrium. So the equality is called as the Gibbs-Donan equilibrium, okay, clear? So I hope it's clear. So this is the equilibrium, okay. So the total cation anions are equal, that is equilibrium. But because of the indiffusible ions not able to move, the diffusible ions are achieving a different distribution again. So that is the case. So let me show it, okay. So it is basically something like this, okay. So first is, this is cations and anions, okay, cations and anions, okay. Or rather I'll write ICF ECF here, ICF ECF, cations and anions, okay. So we'll start the discussion. Now first thing that we have to understand, and we have to see is, okay, fine. So, okay, so we are going with this question, okay, and one thing is first, what is the idea? It is mainly due to which ions? Indiffusible ions. Are you clear? And we are seeing which is the main ion out of these, okay, clear? Okay, fine. Answer, a large number of people already know the answer, okay. Here we are basically discussing why the answers are right or wrong, okay. So answer is obviously given in all books and we have discussed in class also. So what are the cations? Okay, so first is sodium calcium cannot enter, so that's why they stay outside. So they are not able to move, so they stay outside. Why? Because they are indiffusible, so they will stay outside. Are you clear now? And in anions we have the proteins and phosphate, so they again are staying where? Inside, so they are indiffusible, they cannot diffuse. Are you clear? So that's why they are forced to stay on one side. Are you clear now? On the other hand, the diffusible ions are going to be there, okay. This which one? It is our which one? Potassium, and here we are having chloride, so they are diffusible, okay. So technically they should diffuse and they do diffuse, and that's why they create the RMP. So it's their diffusion continuously which create the RMP. Are you clear now? So RMP is caused by this, okay, but what is maintaining the equilibrium? It is the presence of indiffusible ions, otherwise there would be what indifference in the total number of ions. Are you clear now? Okay, so question is, out of the sodium calcium which is more? Now understand, sodium calcium can still diffuse when there is an excitation via the voltage gated sodium channels or calcium channel, or even phosphates have transporter, but proteins have almost no transportation possible. So proteins are having almost minimum transport. So therefore yes, there is some transport of proteins via the vesicular transport, but it's so minimum you can't even put. Like liver is the one making most of the plasma protein, and it is the one who's doing the active transport of proteins, but rest of the cells, especially the neurons, etc., the proteins are remaining almost indiffusible. So that's why out of these all proteins are not going to diffuse much, so that's why they are a stable form of the indiffusible ions which are there in the cell. So that is why they become the main source here. So they ask in the exam, who is the main? The answer is proteins are the main indiffusible ions responsible for Gibbs-Donan. Are you clear? So concept is clear. So RMP caused by, if I want to write, I'll just put it down. So RMP is caused by what? RMP. RMP is by potassium, and also, so it's a major role, minor, okay. So again I'll write RMP, but minor role. Here RMP main role, okay. Then sodium calcium, they cause excitation, they are for excitation. When they come, they cause excitation, depolarization, so they cause the depolarization, okay. And sodium is the one responsible for action potential, so it is for action potential, okay. Calcium can also cause AP, but where? In case of nodal tissue. But if they ask what is the AP, basically we are nodes. Clear? Phosphate are again maintaining the balance, but if they ask you what is the Gibbs-Donan, so proteins are basically responsible for Gibbs-Donan, so they are for Gibbs, okay. So technically they ask what is Gibbs for? Gibbs is protein, phosphate, sodium, calcium, all. Who's the main? Answer is protein, because they're not moving at all. They can move and create AP, RMP. Now is the concept clear? So the answer would be what? The answer is proteins. So they are the, yes, reason for Gibbs-Donan effect, okay. Okay. See, sodium ions have no role in RMP. Okay, okay, Rupesh. Why? Because they are first having almost zero permeability at rest. So therefore they are indiffusible. So they are not moving at rest. So that's why they have no role in RMP. So sodium, no role in RMP. So let me write, okay. So sodium has no role in RMP. Okay. Why? Because they are almost impermeable, okay, almost zero permeability. Everything is clear now, okay. So that is the case, okay, fine. Okay, now we see the next question, okay. Now in our examiner, so now let me first give an idea, okay, who recorded first human EEG. So we know it's Hans Berger, Hans Berger, not Berger, Hans Berger, okay, Hans Berger, okay. And examiner, he give a hint, and say name rhyme with what you get at McDonald's. Okay, we all know at McDonald's we get the burger. So burger rhymes with burger. That means there's a connection in the brain between the two, you can say, word burger and burger. Okay, that means he is trying to activate the memory using a similar sounding this. Okay, now what is the name of this mechanism and how? So before explaining, let me give an example to you people. Okay, let's do an experiment. Okay, all of you, if you can participate with me. Okay, okay, question is for you people. Okay, question is, say the word white 5 times. Okay, so please say the word 5. Okay, say all of you. Okay, 5 times, or we can do 10 times. So say the word white, 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 white. All of you are saying white, 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 white. Okay, now question, what do cows drink? What do cows drink? गाय क्या पीती है? What do cow drink? So what is the answer? It's not milk. Okay, it is water. बात करी उससे कौन किस वाले कर दे जो निकालना पड़ेगा ये इससे करना पड़ेगा इसका एक बार को देखो स्ट्रीम की क्वालिटी कम कर दे कभी 3 से चल रहा है आज का तो जियो वेब से चल रहा है देख तो बैकअप में ऐसे चल रहा है माइक okay. Okay. Iron, sodium. Should we start? I think I'm audible and visible. Okay, so I'll start now. Okay. Okay, so first thing is, don't worry about anything missing. Okay, I'll just do the remaining answers once again. Okay, rapidly. Okay, but problem is, again, now we are in an age of I think everything online. Okay, digital age. Okay, so therefore these things will be a part and parcel. Okay, and this is not that funny. Okay, I've heard stories of people exam getting struck. Okay, clear, and there's nothing on the screen. Okay, that is, I think, more horrific than a class getting struck. So, like, I was a bit tense and why the class not going, but imagine the exam center, the computer freezing. Okay, clear, and it occurred to me also. Okay, because once I gave the exam, it's a different story. Okay, but just to check whether, because when I got my seat at that time, they were just paper pen. Okay, that OMR sheet pencil, you darken the circle. Okay, but I just gave exam NEET PG 2 years back just to see the question. Okay, so at that time, yes, my computer froze. Okay, I thought, what's the problem? Okay, so let's come back and discuss. Okay, fine. Okay, so we were having a discussion on the cow. Okay, so I was giving that idea. So I'll just recap. Here's all the ink. Okay, fine. Okay. So let's start without wasting time. Already we lost around 20 minutes. Okay, 20-25. So as I was saying, the memory which is formed with linking of two stimuli is called as priming. Okay, clear? So priming is basically very simple. The moment I say, let's say I say some word like O, comma, G, comma, D, and I ask you to rearrange. Okay, I ask you to arrange into word. Okay, so what do you arrange into word? Okay, some people will arrange into God, some will arrange into dog. Okay, clear? So you can arrange at God also and dog. So this is an example of priming. Okay, you already had a word in your brain, God or dog, and that's why you're able to reassemble into this. This is called as priming. So are you clear? So priming is nothing but it is, yes, how a previous stimulus affect your response, that is called as priming. So let me now first explain everything, then we will discuss this concept of priming. But you got the idea that you can influence someone's responses or recall by giving a stimulus which is connected to the previous stimulus. So that is called as priming. So let me explain first everything from the beginning. So first is what are the types of memory, etc. So remember, memory we can classify into two categories. One is short-term memory and long-term. Short-term and long-term. Okay. In short-term, basically we have something called as a working memory. Working memory. Okay. And it is for seconds, sometime minutes. Okay. Example, OTP. Okay, so example is OTP. So let's say you get a OTP for your some device. Okay, so when looking at the OTP 46819, the moment you go to type it in the main you can say app page, you forget. So that is an example of short-term memory. So short-term memory we tend to forget very fast. Why? Because it is nothing but neurotransmitter and receptor. Once they are reuptaken, the information is gone from the brain. I've discussed that already. So example is OTP. Okay, so OTP, it is forgotten very fast, very soon. Okay, so it is forgotten very soon, the OTP, and after some time you cannot remember what the OTP was. This is an example of short-term working memory. Okay, and what are the area which process is mainly processed by, so short-term memory processing is frontal lobe. MCQ, if they ask in the exam, it is not stored, it is not stored, working, it is not stored. Okay, it is processed. Okay, then what converts short term into long term? It is mainly conversion. If they ask in exam, CA1 neurons hippocampus. Okay, so CA1 neurons of hippocampus convert short term memory into the long term memory. Are you clear now? Okay, clear? So this is hippocampus, short-term memory into the long-term memory. Are you clear? So short-term into the long-term memory. Okay, so short-term into long-term memory. Okay, clear? Long-term memory is basically stored in neocortex, mainly neocortex. Okay, which lobe? Basically medial temporal lobe. Are you clear? So they ask which area? Medial. Who's the converter? Hippocampus. Okay. Now one more question is, what is the mechanism, the synaptic plasticity involved? So remember, the synaptic plasticity involved here is post-tetanic potentiation. They're all exam question, okay? Post-tetanic potentiation. Are you clear? So it's post-tetanic potentiation. Whereas in this case, the synaptic plasticity is, okay, that is PTP. And here for long-term memory, it is LTP and LTD. LTP and LTD. That is long-term potentiation and long-term depression. Are you clear? So long-term potentiation and depression. Potentiation is excitatory. It's mainly used in glutamate, NMDA receptors. Are you clear? So it uses NMDA receptors. Are you clear? So NMDA receptors. Okay, fine. Whereas long-term depression, so long-term depression is basically be having what? It is basically GABA and GABA receptors. Are you clear? So this is the summary we have got. Okay, so mechanisms are clear. Okay, how they work I've discussed that already multiple times, that when you increase the receptors, the synapses which are involved in that processing of data become more activated, and in future you're able to recall and retain the information much better. That is called as memory formation. So summary: long-term memory, neocortex. Which area mainly? Medial temporal lobe. What is the converter of short-term to long-term? Hippocampus. CA1 neurons. What is the mechanism mainly? LTP and LTD. What is LTP? Potentiation. What is LTD? Depression. Are you clear now? So that is a summary. Okay. Now let's see the other important concepts. Okay. So let me just show. Okay. So question is, what do we have or what do we see in this case? Okay. So please write down. Okay. So first is, right, the types of long-term memory. Okay. Types. Okay. So basically they divide into the two types. Okay. Implicit and explicit. And implicit. Are you clear? Okay. So implicit and explicit. Okay. So implicit and explicit. Now what do you mean by the term implicit and what do you mean by the term explicit? Okay. So implicit and explicit. Okay. So let me explain. So first is the implicit and the explicit. Implicit is also called as non-declarative. Okay, or you can say it is unconscious. It is basically unconscious. Unconscious recall and memory. So what you cannot recall back consciously, that is there in the brain but you can't use it, so that is called as the unconscious things. Okay, so that is not fully. Explicit is expressive memory. You can bring it out consciously. Okay, so it is declarative. Declarative. Okay, that is conscious. So we can immediately, yes, so it is, yes, we are aware of this and we know what this information is. Implicit, we are not aware, it is already there and we can just use it, but we don't know what it is. Okay, and what is it there. Okay, so let's start the discussion. Okay, so discussion will start with explicit. So it is two types. Okay, it's two types. One is semantic and episodic. Are you clear? And the episodic are the types. Okay, so semantic and episodic. Okay, so remember what is the semantic and the episodic are and what is the funda. Okay, so I hope everything is fine. Okay, so what is the funda in this particular case? Okay, so first is, remember the case is, okay, basically episodic is episode events is basically about the events. Okay, like you go to a birthday party, so remember the cakes were there, the cake was very big. Okay, or it's about the information. Okay, semantic is basically the information about the self. Okay, so it's basically what it is, the memory about self, so also called as autobiographical memory, autobiographical. Okay, and this is events memory. Okay, so semantic is the one related to your own life, like your name, address, relatives. Okay, or what your education, your school, all this information is basically semantic. Are you clear now? So semantic is basically about all the information or facts related to your own life. Okay, so mainly facts. Okay, clear. Whereas episodic is event memory. Okay, like example, you go to a birthday party or you see some news. Okay, so in this case the everything like news, okay, birthday party, or some cricket match, or all these things, so they are going to be episodic thing. Okay, clear. And what is it stored? Obviously it is stored in the temporal lobe, which I discussed already, and you're able to recall it very well. Okay, so that is explicit memory. Okay, clear. Now we will discuss basically the implicit memory in this particular case. Okay, so what is the implicit memory types and this? Okay, so first is, let me explain, it is basically having, again space is the issue because, okay, so let me do one thing. I'll use OneNote because OneNote has this ability to scroll down. Okay, so I'll be using OneNote for explanation. Otherwise, I'll keep deleting and adding info. So, yes. Okay. So let me discuss. So I'm discussing what now? The implicit memory. Okay. Now, implicit can be divided into various category. One is your procedural memory. Okay, or skill memory. Are you clear? So procedural or the skill memory is the term. Okay, clear. So procedural or skill. Okay. Now first example is driving, writing. So therefore we don't really actually know that when you're writing, do you think A is go up and down, then you just make A. You're not even aware. Or when you're driving, you change the gear and you don't even have to consciously be aware ki, okay, first gear, second gear, you just change the gear and drive the car. So that is the example of driving or this. Okay, or even when you walk, you have a habit of what, like touching your nose, or your hand goes like this. Okay, so you're not even aware which hand is going front, back. So some people walk with, so all their walking manners, etc., they become, so even your manners or habits, they become a part of it. Like your habit of touching let's say someone's feet, so don't think the moment someone come you touch, like your notebook fall down, immediately pick and put it to your head and you touch like this. So this is not something you do after thinking conscious, it's just a behavior. So that's why all these things are called as procedural skills, or sometimes you also use a very important word like motor memory. Motor memory. Okay, and what are the best area for this? So best area involved for these is cerebellum, also basal ganglia. Clear. Also they are also involved in premotor cortex, so they also be involved with premotor area. So these are the three areas which are mainly involved in all this habits, skills, all this. Okay, clear. So that is cerebellum, etc. Okay, fine. Okay, now the other two types that we see or have is, number two is conditioning. Okay, now conditioning we know is basically classical and this Skinner. Okay, so conditioning I'll discuss just some time. Okay, conditioning, and the next is priming. Okay, clear. Priming. Okay, so what are the three? Let me revise: skill memory, conditioning, and priming. Okay, and priming. Okay, clear. Now question is, some books also explain that conditioning, like example we know. Okay, so food, saliva. So when you see food, the saliva is there in the mouth. Okay, so when you see the food, the saliva is there in the mouth, and that is called as what? That is called as a conditioning. Okay, so saliva causing food is conditioning. Okay, now you know, if you combine food plus bell, what happens? Eventually the bell only cause saliva. Like example, we are taught something. Okay, Like let's say when you enter your classroom, what happened? When you enter the classroom, you are, let's say, doing something. Okay, so when you enter the classroom, you wish the teacher, or so now what happened? The moment teacher come, you immediately stand up. So now what happens is it's a kind of conditioned response. Okay, that whenever there's an action, you do a response. Okay, and that is based on your previous memory. Okay, clear? So food plus bell cause saliva, eventually the bell. This is called as classical conditioning and was done by Pavlov, also called as Pavlovian. So the dog now can make the saliva, not with food. Food with saliva is a reflex. Food with saliva is a reflex, and bell with saliva is conditioning. Okay, clear? Whereas food causing saliva, like your hand touch a hot object, you withdraw. This is example of what? This is an example of, yes, reflex. So it is basically a innate reflex. Innate reflex. Okay, and this is a conditioned reflex. So now it is a conditioned reflex. Easy. So this is basically a conditioned reflex. Okay, so stimulus causing a response which was not earlier is called as conditioning. Are you clear now? So earlier bell never used to cause saliva, so bell no saliva. Now bell cause saliva is called as conditioning. Are you clear now? So it is conditioning. Okay, so these are the two. Now we see priming. Okay, now what happens in priming? So again in priming, a previous stimulus, okay, affects the response based on its connection with the another stimulus. Okay, so in this case, a stimulus, previous stimulus, affect the response to current stimulus. Are you clear? Like the example I gave you, when you say the word milk, white, 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 your brain, all the neurons connected to the word white start firing. Like white is milk, clear? White is the moon, white is okay, the color of peace. So now all these neurons are primed the moment you give a stimulus of cow. So cow has its own connection. So cow gives milk. So white is also connected to milk. So milk neurons become the most active, and you immediately speak the word milk. And that is example of what? Priming. Are you clear now? It's an example of priming. Okay. So that is why when you eat burger, okay, in the McDonald's, you eat burger. So this burger is also the similar name to the scientist Hans Berger. So Berger triggered by burger triggering Berger rather, is example of what? Priming. So therefore the previous stimulus, okay, affect the response to current stimulus. So that's why what he asked you about the EG scientist. Okay, so EG scientist name is Berger, and the previous stimulus, what you eat at McDonald's, will give you the ability to recall. So that is called as priming. Are you clear now? So priming is the example, like example we call. Okay, so this EG, okay, Berger and McDonald's, Berger, so they get connected. This is an example of priming. Are you clear now? So answer would be obviously in this case. So what is the answer for this question? So answer is priming. Okay, why not reflex? In reflex, the stimulus cause a response. Like the teacher is, let's say, doing something. So the moment you see, so sorry sir, sorry. So the moment you see a teacher and you say sorry, or by mistake you touch the teacher, and your shoulder touch, you say sorry. That's a reflex. Okay, clear? So immediately you say reflex. Associated learning again is basically you are associating. A for apple, B for ball, they are all declarative memory. No, it is not declarative, it is an implicit memory example. It is implicit, not explicit. Okay, once you recall, like example I ask you what is your name, what do you eat at McDonald's? Eat at McDonald's, it is obviously Berger. This is explicit, declarative. But if they ask you what is the mechanism which is helping the student recall, that is priming. Are you clear now? So that is priming in this case. So obviously it is not associated, it is, yes, it is declarative. What is declarative? The ability to recall that burgers are given at McDonald's. This is example of declarative memory. But is he using this as the mechanism? No. Mechanism is priming in this case. Are you clear? So priming is the answer. Okay. Fine. So let's see the next question. Calculate the ECF. So first question, let me just revise. Okay, so what is the formula for volume? So volume is given as amount of dye minus, okay, excreted amount divided by the concentration of dye in the chamber. So we have done that already. So amount of dye minus excreted value divided by the concentration give you the volume. So it gives you the volume. Are you clear? So it gives you the volume. Okay. So let's solve it. Okay. So first is what is the amount of dye? They say it is 10 gram. So since it is in milligrams, so what I'll do, I'll write in milligrams. Okay. So 10,000 milligram. Are you clear? So 10 gram I've converted to 10,000. Excreted is 10%. So that is around 1000 milligram. It's 10%. No? So 10% is 1000. So 10% is 1000. Okay. Clear? And divide by the concentration, that is 65 milligram per 100 ml. Are you clear? So 65 milligram per 100 ml. Okay. So if you solve it, what do you get? So if you solve, you will get something like this. Okay. It is 9000 milligram by 65 milligram, and 100 goes up into 100 ml. Okay. Fine. So let's solve it. Okay, I know it's a bit. Okay, so what we can do is we can use a calculator. So milligram is cut. That is the first thing we should do. Okay, so milligram is cut. Okay, now I can solve it by this also, but better I'll use a calculator. Okay, so okay, why waste time? Okay, so let's use a calculator. Okay, so what was the value? Yes, it is 9000. Okay, so 9,000, rather 90 is okay, divide by 65. So get what? 13.864. Okay, 86 liter. Okay, why? Because ml I convert. Okay, is actually 13864 something. So if you round off, you can write 13.86 liter. Okay, roughly we can take 14 liter. Okay, clear? So I don't know why they gave such tricky. They could have given an easier calculation. Okay, but again, fine. If they've given, let's solve. Okay, so 14 liter is the ECF volume, because we use mannitol for ECF. So mannitol is used for ECF. Okay, clear? So the answer is yes, around 14 liters. Okay. So again, it's a repeat question. Okay, asked 2 years back in the exam. In fact, most of the questions you can actually, if you do the previous question, you can solve very easily. Okay, fine. Okay. Next, what is not required for calculation of creatinine clearance? Again, I've discussed clearance multiple times, and I discussed U V by P. Are you clear? There's a formula for clearance, U V by P. Okay. In fact, I also gave in the class the formula for calculation of clearance. Okay. So what is U here? So U is urine concentration. So what is U? Urine concentration. So if you are doing inulin clearance, it's the urine concentration of inulin. P is plasma concentration. And what is V? It is volume of urine per minute. Now that means you need to know the volume of urine that is formed every minute. Okay. So how do you get this? Okay. So how do you get this volume of urine every minute? Okay. Fine. So let me start. Okay. So idea is very simple. Okay. So urine. So how you do? First we take a person, and why creatinine clearance is preferred? The answer is very simple. Creatinine is already in the body, whereas inulin has to be given from the outside. So therefore, if you are doing creatinine, the easiest thing is what? First, we take a urine sample for urine concentration. We need the plasma, that's blood sample for concentration in the plasma. And finally, volume of urine. Now you cannot ask the patient to do something where you can check per minute. So what is it given by? So it is basically 24 hours urine collection, which I discussed in the class itself. And I also gave this example. Babaji, you give a box and ask Babaji to collect the urine in this container which has marking, so that you can check what is the amount. Although it doesn't look like this. Okay, it's very narrow to you understood. Okay, so you cannot use this bottle, so you give this proper urine, you can say containers where the patient urinate, and you check the amount of the urine he or she is passing, okay? And make sure giving the instruction clearly, okay? Like collect only your urine in this, okay? Okay, you don't know patient, okay? They might be collecting urine of everyone. Hey, you pass urine only in this, the doctor said. No, it's your urine, not everyone, okay? And second, they sometimes don't even understand. Like once we give instruction to a old man from the village that you bring the urine next time for testing, okay? So we were suspecting some, I think prosthetic or something, okay? So next time we come to the OPD after a week and he was bringing one container, you know, that ghee, the one you contain the ghee, okay? So thought he might have brought ghee from the village, and then he said, "Lo, doctor, do the urine test." It was filled with urine for his past so many days, okay? So therefore, yes, 24 hours urine collection, okay? So let me give an idea how this thing works, okay? So let's say in 24 hours he make 2400 ml urine. So how much urine? 2400 ml urine is made, okay? So that means in 1 hour how much is he making? 100 ml. Are you clear? So he is making 100 ml. So in 60 minutes how much will it be? It will be 100 ml, or in 1 minute it would be 100 by 60, that is 1.66 ml. Are you clear? So 1.66, and that gives us a V equal to 1.66 ml per minute. Are you now clear? So if you multiply these three values, you will get you will get the value, okay? So these three value you will use for getting the clearance, okay? So you don't need the amount of plasma filtered. Why? Because after filtration you do reabsorption and secretion also, so that's why it is not a value we need in this case, okay? What are the amount of plasma filtered or going through is 180, we know, but is not required for urine formation, okay? Yes, it does affect the urine formation, but it's not the only process. So what is the right answer? Right answer is we need this, and we don't need this, so this becomes the answer. So D is the answer in this case, okay? So many D's are the answers, okay? Fine. Now let's move to the next question. In skeletal muscle, DHPR and RYR. Okay, now again I've discussed that multiple times in the class. Okay, so what is DHPR? It is dihydropyridine receptor. Okay, it's basically a voltage gated calcium channel, also called as L type. So it's a voltage gated calcium channel, okay, dihydro. So therefore this allow the calcium movement, okay? So it's a basically a electrical channel, okay? So it's basically electrical. Is basically electrical, okay? Clear? And it is coupled electrically. It is coupled, okay, to RYR on sarcoplasmic reticulum, okay? And RYR is nothing but a channel to allow calcium to come. So what is RY? It's ryanodine. So RYR is the ryanodine receptor. Are you clear? So it is the ryanodine receptor. Let me show the diagram, okay? So this is the case, okay? So this is RYR, that's ryanodine, and DHPR is dihydropyridine, okay? And STAC, STAC, okay? The full form is, it's something like skeletal muscle T. So it is basically a protein which couple the two. So it is said to, it help in the connection, okay? So it is for coupling. So STAC, okay? So the STAC 3 gene make the protein stacks, and this is the one which help in connection, okay? Clear? So therefore what happens is when the action potential AP, when the AP reaches the sarcolemma, so this thing which you see is sarcolemma. So let me use a color change. So this is the sarcolemma. So the action potential will travel, and via the T tubule, which is a fold in sarcolemma, it is able to excite the dihydropyridine. You can see the sign volt. It is electrical in nature. So therefore, there is an electrical excitation of this. This is the calcium channel. So you can see calcium. So once this calcium channels are activated, they will activate the RYR. There is ryanodine. Once ryanodine is open, the calcium is released. So that's why it's like a door. When you open the door, like this bottle, if I open the mouth, the water comes. So this cap is a mechanical device which on opening releases the water. So this RYR is a, you can say, receptor which allow calcium to come. So technically it is a what mechanism through which calcium comes out. So it is what, a mechanical, you can say, thing. Okay, so it is mechanical in nature. So this is mechanical. So mechanical release calcium, release of calcium. So from the sarcoplasmic reticulum, the calcium get released into this and then it causes the contraction. So that is why technically it is electrical then mechanical, electromechanical coupling, where stack 3 play a key role in this thing. Okay, clear? So stack 3. Okay, fine. So therefore, any deformity in the stack or RYR can cause problem. Like example, it is said or hypothesized that RYR or stack 3 defects mutation are responsible for something called as malignant hyperthermia. So the clinical correlation, okay. So a mutation, okay, in stack 3, stack C3, RYR gene, okay, stack 3 RYR, okay, produce, okay, malignant hyperthermia. Okay, so it can produce what? Produce malignant hyperthermia, where the muscle continue contracting and doesn't relax. And who causes? Is caused by muscle relaxant like the succinylcholine, okay, like the depolarizing ones, or also by the anesthetic agents. So therefore malignant hyperthermia is due to, yes, muscle failing to relax and keep on contracting due to too much calcium causing continuous contraction. That is the pathological concept which is involved here. Are you clear? Okay, so what is the answer in this case? So first, it is GHPR, it is electrical. What is RYR? It's a mechanical system for release of the calcium. So it is becoming what? A electromechanical. So electrical followed by mechanical. Okay, so electrical followed by mechanical. Okay, so this case. Okay, now this graph shows the normal rate of synthesis. So now as you can see, okay, first there is something called as this first one. So first one remain almost half. That means 50% are the these chains which are remaining consistent. Okay, B are almost same as the A, that is almost 50%, but then it decreases and become hardly 2 or 3%. So that means if I start labeling, this is 50% throughout from the beginning. So what time it start? Third week. So we know that hematopoiesis it start at third week. Okay, third week yolk sac. Okay, so hematopoiesis start at the third week in the yolk sac. Okay, clear? And therefore what is happening? There is the case. Okay, now first it is 50% throughout from beginning to the, you can say, 6 months. That is in the infant it is 50%. The B1 is 50%, almost 50% in the beginning, and then now it is hardly 2 or 1%. Okay, so you can say just it is 1 to 2%. So B is reduced. Okay, so it is 50%. Then it is this is A. A is throughout. This is the option B, which is decrease. Okay, fine. Then comes the this one. Okay, the C ones. Okay, so what happens in the C? C is almost 1 or 2% in the beginning, and then it keep rising, and now it is 50% in this case. So this is 50% towards this age, 50% here. Okay, so 50% in the after birth phase. Okay, clear? So what could these be? So let me explain. We know that a fetus is having various hemoglobin. Okay, so we have fetal hemoglobin. So let me explain first. Fetal hemoglobin, it has what chains? HbF, we know, 2 alpha and 2 gamma chains, 2 alpha and 2 gamma chains. Okay, clear? Fine. And adult has 2 hemoglobins. So adult hemoglobin HbA. Okay, so adult HbA has 2 alpha and 2 beta chains. Okay, 2 beta chains. Okay, clear? That is 95%. Okay, 5% or less than 5% are the other ones. Okay, so question is, that means what happening? The gamma chains will decrease after birth and the beta chains will increase after the birth. So that means alpha chain are 50% in both adult and the fetus. So that means this must be the alpha chain. So this is alpha chain. So this must be the alpha chain. The green one, which is rising, becoming almost 50%, and you know it is 95% of the hemoglobin in adults. A1 and A2 will discuss in brief soon. Okay, so this must be the which chain? This one must be the beta chains. Okay, and this one which is almost disappear must be the gamma chain. Are you now clear? So we have got the answer that gamma chain must be the one which is in the orange one. That is the case. Okay, fine. Now what about the other chains? So they have already mentioned this is delta. So what delta is? They are after birth, slightly before birth, but they are very low. Why? Because it is very little in amount. So let me explain, because space is not there. So you know, HbA2 contain 2 alpha and 2 delta chains. 2 delta chains. Okay, clear? And it's hardly less than 5%. So it's less than 5% in adults. Okay, and this is 95%. So therefore in adults 95% is adult hemoglobin and just less than 5% is the HbA2. So this must be 5% which we are having. This is this, and what is just 1 to 2%? It is this. So that means fetus has HbF mainly, but adults have HbA mainly. So therefore in adult, what is the amount of HbF? It is hardly 1 to 2%. So one more thing to remember is, okay, adult have less than 2% HbF. Remember this statement. So adult have less than 2% HbF. But in case a adult cannot make the beta chain, to compensate they will now make more and more the which chains? The gamma chains, and therefore and also delta. So therefore their amount of HbF increase. So therefore HbF can increase in adult in thalassemia. Okay, beta thalassemia. So in beta thalassemia there is increased HbF more than 2%. Okay, fine. Now, what about this D? So this D is basically seen in the fetus, okay, embryo, okay. So therefore in embryonal stage, basically this is embryonal hemoglobin, okay, and primarily they contain what? This embryonal hemoglobin contain which chains? They contain the E embryo E epsilon. So they contain the epsilon chain. So they contain the epsilon chain. Epsilon chain. Okay, so what is the content? They contain the epsilon chain E. Okay, so let me just show them and then we'll discuss. Okay, so adult is main is A2, which is alpha 2 beta 2. A2 is alpha 2 and delta 2. F is alpha 2 and gamma 2. Okay, which is less than 2% adult. Okay, embryonal is basically Portland, they don't ask much. Okay, it is the one which is formed the first. Okay, you can just skip. The main is this one. Okay, so it is epsilon, oh sorry, it is zeta 2 and gamma 2. Okay, zeta, okay, this is zeta. Okay, zeta and this. Okay, and gover 1 and gover 2, they contain basically which one? So gover 1 contain the, yes, epsilon, and it contain the zeta and the alpha. Okay, clear? And finally abnormals are when there is what? All chains are beta, it is called as H, and BART is all are gamma chains. Okay, they are the abnormal ones. Okay, so therefore what you remember is these ones. Okay, adult, fetal, and here mainly you remember this govers. Okay, fine. So let me just show them. Okay, so this is the case. So first in yolk sac, what start to form? The epsilon and the zeta. So this is the embryonal hemoglobins. Okay, both Portland and the gover. Are you clear? And they disappear within, you can say, 8 or 9 weeks. Okay, fine. Then the beta is starting afterwards, and beta will be low in the you can say fetus, and it's actually starting after the birth. It exceed the amount of the gamma chains. So gamma chain, this is HbF, which is the case. Okay, so HbF is using, and this is HbA. Are you clear? Fine. Delta here is HbA. Fine. So this is, I think, a very simple summary where you can remember all the cases. Okay, clear? So anyone who want this graphs, etc. Okay, so I will send this PPTs. Okay, either on mail or you can say I'll give it on Telegram, etc. Okay, so you can just message me there and I will send it to you. Okay, clear? So Telegram, or you can say mail. So I'll give this PPTs. Okay, which contain all these explanations. Okay, so number is 9899, and you can also SMS in case you're not using Telegram. Then I will tell you the link of the Google Drive. So on SMS I'll give you the drive link. Okay, so drive link on SMS in case you're not having Telegram. Okay, or mail is Ashish mamc@rediffmail.com. So rediffmail.com. So you can get this thing there. Okay, fine. So I hope it's clear. Okay. So the answer for this question is which one? Yes. So this is basically which hemoglobin? So they are asking gamma chain. So gamma chain must be C. So answer is C. Okay. So gamma, sorry. So this is not C, but it is which one? B. So B is the answer. Okay. So B is the gamma chains which are decreasing. Okay, fine. Which is decreasing. Okay, fine. Okay, so B is the answer. Okay. Okay, fine. Now question, male patient present. Now obviously you can see the BP is very high. Okay, and when you see in this BP something is about pulse pressure. Okay, so we know pulse pressure is SBP minus DBP, and here it is around 190 minus 100, sorry, minus 100 is 90 mmHg. It's very high, almost double the normal value. Okay, so very high pulse pressure. Okay, so first question is, what are the reason for this high pulse pressure with age and things? So first understand what the pulse pressure basically indicate. It basically indicate stroke volume. Stroke volume. Okay, that is the amount pumped by the heart in every beat. Okay, so that is stroke volume. Okay, clear? So stroke volume it indicate. Okay, fine. So therefore, if there is more ventricular contraction, more the stroke volume, more the pulse pressure. So again, stroke volume will increase the pulse pressure. Cardiac output, as heart is increasing, more cardiac output most commonly, if not changing the heart rate. If heart rate is not changed, then again stroke volume is the main, and myocardial contractility will increase the stroke volume. So that means again, so these three will increase the pulse pressure. So they will increase the pulse pressure because they are all basically increasing the pumping. As heart pump more blood, there's a massive increase in the cardiac output, stroke volume, and therefore systolic BP will rise, and also pulse pressure tend to rise up. Okay, fine. Now one question there you have to understand is, what is the relation with the arterial wall compliance? Okay, now understand something very important. Okay, there is something called as elastic recoil. Okay, elastic recoil, mainly aorta and arteries, aorta and elastic arteries. Okay, now what is the elastic recoil do? It basically produce the diastolic blood pressure. Okay, so diastolic blood pressure is produced by the elastic recoil in this particular case. Okay, now understand what happens is, as there is blood entering the aorta stretches, the aorta will stretch, and now it will recoil in diastole, and that produce the diastolic blood pressure. Okay, so just to give a brief idea in this case. Okay, so therefore what happens, let me show. Okay, so understand. Okay, so what happens is, this is ventricle, this is aorta. Okay, so so this is the left ventricle. So as the left ventricle contract, it produces the systolic blood pressure. So it produces the pressure of 120 mmHg. This is aorta, and it will force blood into the aorta with a pressure of 120 mmHg. Okay, and this will then cause the systolic flow. Okay, and also the main cause of pulse pressure. Now this causes the aorta to stretch up. So aorta will stretch. Are you clear? So aorta is stretch. Are you clear? So this causes the case. Okay, so as the aorta is stretched, it causes the blood to flow, and also that means save the energy. Okay, that is systole. So this is all occurring in systole. Are you clear? Now in diastole the story changes. In diastole, first thing, the ventricles are no longer recoiling but they are relaxing. So they are going outward, they are relaxing. So this is diastole now. So now what happens, the pressure of ventricle fall and can fall almost zero. So the pressure now almost zero. Okay. Now the aorta which was in a stretch condition will now recoil. So aorta and the elastic vessels recoil. So aorta recoil. Okay. Now as it recoil, it will compress the blood inside. Compress blood, and as the blood is compressed, the pressure becomes 80. Okay, not 100. Why? Because some energy is lost as inelastic work. Okay, so therefore, and this causes diastolic flow. Okay, and some blood will try to go back, and that is prevented by closure of semilunar valve close, and that causes dichrotic notch. So therefore the closure of the semilunar valve will prevent the blood from going back to the ventricles. Are you clear? So this is the DBP. Are you clear now? Okay, so therefore we get both the pressure 120 and 80. Now what happens, in old age the aorta becomes stiff. So in old age there is aortic stiffness. Okay, so aorta becomes, so aortic compliance is reduced, and that is called as aortosclerosis or rather arterio. So not just aorta but even elastic artery is reduced. Okay, and they become stiff. Okay, and that is called as aortosclerosis, or if arteries, arteriosclerosis. Are you clear? So with age there is a reduced compliance. Okay, and now they are not able to stretch. Are you clear now? So they are not able to stretch much. Okay, clear? So they are stiff. That means stiff, they can't stretch. So now the result is what? As aorta cannot stretch, the ventricle, when it's pumping blood, has to pump blood with a greater pressure to compensate for stiff aorta. So therefore there's an increase SBP due to stiff this. Okay, so increase SBP. Okay, so SBP will rise in old age. Okay, and since aorta doesn't recoil, it's stiff, it doesn't recoil, and there is decrease DBP due to poor recoil. So DBP will decrease, and the result is this cause a massive increase in pulse pressure. Why? Because of rising SBP, the ventricles are working hard, and a falling DBP. So that is why it is causing all these changes. Are you clear? So therefore, pulse pressure will increase. Pulse pressure will increase with age because of stiffening of aorta and arteries, and this is called as sclerosis, arterial aortosclerosis. Okay. So therefore, what happens? Okay, as there is a compliance is decreased, there is a increased pulse pressure. So therefore, as this compliance decrease, it causes the increased pulse pressure. So what they are asking in this question, they are asking which parameter should decrease for this high pulse pressure. So answer is arterial compliance decrease makes the aorta stiff, artery stiff, causing high systolic pressure and reduce. So therefore, stroke volume, this all three will increase the pulse pressure. On increasing, the only one decreasing, answer is compliance. So we have discussed that in the class also. So that is the question answer. Okay, so answer is what? Answer is A. Okay, so answer is A. Fine. So answer is A. Okay, fine. Question number 10. Okay, a single neuron receive 500 inputs from 150 different sensory neurons. Okay, now this thing that you have to understand is there is something called as convergence and divergence. Okay, so therefore convergence is they all so multiple inputs to one uh, one neuron. And what is divergence? It is multiple outputs from one neuron. Okay, so therefore there are two techniques, convergence and divergence. Okay, so let me first explain why we have feed forward feedback is not involved. Why? Because they have to go back. Okay, in this case, yes, it can be feed forward, but again it is not the right answer. Why? Because they are saying different sensory cells. Okay, so once they are different sensory cell, and that means what, some are giving 3, some are giving 4, some might be giving more. Okay, so some are giving more input, some are giving less. Okay, and some are inhibitory also, some are excitatory, remember. So it can be both inhibitory or excitatory. So therefore these are wrong options. Okay, feedback is going back, you're not going back, you're going forward, so this is wrong. And it can be both. It is feed forward, I agree, it is feed forward, but can be excitatory also, inhibitory also. It is feed forward but not only excitatory, can be. So therefore the best answer would be convergence. Okay, now what is the significance of convergence and divergence? Let me explain. Okay, see, understand. Okay, imagine this is a single neuron. Okay, clear? This is a single neuron. Okay, clear? And this neuron is ending on, let's say, a muscle. Okay, and this muscle will either contract or not contract. So who decide? This single neuron. Are you clear? And this single neuron receive information from, let's say, 3 different neurons. Okay, 3 different neurons. Okay, why am I making them in different class? So let's say this create inhibitory, so it is inhibitory, called as IPSP. So that's what, this is a feed forward inhibition, it is inhibiting. So the feed forward inhibition, so you are inhibiting, and these two are exciting, so they are excitatory, let's say EPSP. So this is plus and this is minus. Now what happens is, based on their summation, the neuron will either contract or not contract the muscle. So this is a simple diagram showing the convergence and summation. So this is called as convergence and this is summation. Are you clear? So convergence and summation. Okay, so convergence, this is feed forward inhibition, feed forward excitation. So therefore don't be confused, it can both be feed forward stimulation also, feed forward inhibition. Okay, clear? So let's start the discussion. What are the example? Okay, so someone abuse you, he say MCBC something. Okay, MCBC. Okay, clear? So what happens? The neuron should be excited and contract, and you slap the person. Are you clear? So slap. So there is a conversion and summation, you slap the person. Okay, but this person is your HOD, okay, or your SR. So it will inhibit and you don't slap. No, sorry. So you want to slap, but what? So that is what convergence create all the responses. Are you clear? So the sensory input, he is giving you abuse. First he say MC, then he say BC. So it's a very high excitation. You will contract and hit the person. Okay, but your HOD, he is your HOD. So that is inhibitory and you will stay there. Are you clear? So that is convergence and summation. Okay, now what is divergence? Okay, divergence is when one input goes to multiple source. Like example, you see, let's say rows. Okay, don't ask me to make rows. Okay, so this rows information is coming to the sensory neuron. Okay, so sensory neuron is getting this information. Okay, now this sensory neuron is going to give the info to multiple other ones. Okay, so this will give info to other 3. Which 3? Let's say it give the information to olfactory cortex, so you can smell, and now this is the case. Okay, then it can give to visual, so you can see the rows, okay, and maybe it also give to the motor system. And what do? You are now holding, so you try to hold or touch, or you touch the, okay, or rather hold, so you will Yes, take or touch or hold the rose. So therefore, when someone shows a rose and you want to hold or you want to put this muscle causing the button for this, okay, so that is the case. Okay, so therefore you want to, let's say, give a thumbs up. I give something, so therefore whatever I'm saying, it is causing a divergence of information in your brain. You are having multiple, this is called as divergence. So your brain is doing divergence, convergence all the time, and you are having all these responses. Are you clear now? So this rose, I'm trying to make this rose, but I know I'm not capable. So this is the rose, okay, with the green leaves. Okay, fine. So this is the rose you are seeing, and it is creating all these different responses. Are you clear now? Fine. So obviously the answer is what? The answer is convergence is the answer. Okay, because they are all ending on this. Okay, clear? Single neuron. Okay, fine. Now, mechanism of transcutaneous electrical tense. Okay, so tense is used for pain, is for pain management. Okay, is for management of pain. Okay, so just ignore this thing. Okay, so it is for management of pain. What is the mechanism of action? Now, okay, so before explaining, let me explain something called as the lateral inhibition, also called as the gate control theory. It's taught in pharma also, so that's why again it's a question of pharma and physio both. Okay, so gate control theory. Okay, so what is this lateral inhibition or gate control theory is? So therefore what happens, okay, the pain pathway, okay, it is having a convergence. What happens? The large afferent A fibers, so large afferent A fiber, okay, which include mainly A beta, which is for touch, etc. A beta for touch, pressure. Okay, so touch, pressure, they inhibit smaller afferent, mainly C fiber, and A delta also, mainly C fiber in spinal cord. So this is called as the gate control theory. Are you clear? This is called as a gate control theory. Okay, so gate control theory. Okay. So what is the result is, if both pathways are triggered at the same time, the large pathway and the small afferent fiber pathway, that is C, and you know all C carry what? It carry pain. It carry pain. So mainly visceral pain. So therefore, once the pain, and even some books say A delta, which is for fast pain. So therefore, both A delta and the C fiber are suppressed by the larger, because when they enter spinal cord, there is this mechanism. Okay, so let me explain. The A delta and C, which are carrying what? They are the small nerve fibers. They are small fibers. Okay, so they are carrying the pain and all injury, etc. So nociception, that is pain pathway. As they are reaching the transmission cell, okay, these transmission cells are primarily the substantia gelatinosa. So they're basically SG cells. They are the main transmission, that is substantia gelatinosa. Okay, so they are the one who give the pain pathway. So they make the pain pathway. So therefore they will receive the pain pathway. Okay, so now what happened? There is a inhibitory interneuron, and this inhibitory neuron is going to what? It is going to allow whether the pain will go or not go. So now what happens is, once there is activation of the this massage or this by A alpha and E beta, means A beta, okay, large fiber, this will excite the inhibitory neuron. So it will stimulate the, and this inhibitory will now suppress the transmission cell, and therefore what happen? The brain will not receive the pain, but rather it will receive the touch, pressure, etc. So you will feel the massage or the touching or even the electrical signal received by the skin and suppress the pain sensation. So therefore what happens is, therefore the massage or acupressure or even this tense or even something called as this counter irritant like Iodex, Jandubam, okay, they are called as counter irritants. okay, like iodex. So they will suppress the pain by exciting the, yes, A fiber, okay. So the larger A fiber suppress, so that's why they will suppress the pain. So this will suppress pain, and is most effective in the C fiber inhibition because they are the one who are inhibited primarily in this case. This is called as the gate control theory. Are you clear? So inhibitory neurotransmitter, yes, but again it's not the main mechanism. Main mechanism is gating at the spinal cord, which is causing the pain not reaching the brain. Okay, adrenergic is not the case because that is for sympathetic, and sympathetic rather cause the pain. Sympathetic fiber carry the pain, so sympathetic carry pain. So therefore, yes, they carry the visceral pain, not right. Inhibitory not. Supraspinal, that is descending pain pathway. Supraspinal control is true. So there is something called as this para-aqueductal gyrus, which is inhibitory pain. So again, but it is above. It is not used by TENS. TENS is via activation of skin via the electrical currents. They activate the larger A fiber and suppress the pain. So supraspinal is descending pain control. So again, it is not for TENS. It is descending pain control. Okay, so you can ignore. So best answer is gating at the spinal cord. Okay, gate control theory. Okay, fine. So let's start next. Okay, so we have discussed all the questions in this case. Okay, so if any question left or not done, okay, so you can just inform. I will try to give the answer whenever is possible in this particular case. I hope it's clear. So that is the summary we are having. Okay, clear? Fine. Okay. Okay. So the remaining initial answers. So I will just give the ones which might have missed. Okay, so let me just summarize the answer from beginning. Okay, so first one is thyroxine because thyroxine is lipid soluble and lipid soluble hormones directly act through nuclear receptor. So answer is nuclear receptor is the answer in this case. Are you clear? Okay, and I also give the mnemonic dirty pig. Pig is prolactin insulin growth hormone. Then negative feedback, gain is infinity when there is zero error, and zero error is possible in case of kidney because they have perfect control over the volume that they are maintaining in the body. So what they do, maintain perfect volume. So that is the answer. 2, we've done that. Infinity. 3, Gibbs Donan equilibrium is mainly by this indiffusible ions, and which is the main here, proteins are the main indiffusible here. Why? Because they are having almost zero permeability. They don't diffuse at all. Others can still diffuse. So that's why RMP by potassium chloride, AP by sodium, even calcium can cause AP nodal tissue. Phosphate again is not able to do any of the, but yes, it is having again the same effect. Okay, indiffusible ion. Okay. Again, I told you it is priming. It's an example of priming. Okay. So we discussed that already there. Okay. Then ECF volume we have done. Okay, the answer is 14 liter. Here the answer is priming. Okay. So I think we have discussed all. Okay. So thank you very much. Okay. In case any doubt etc., you can either type here. Okay, in the chat, or we'll discuss it later on. Okay. But remember the summary is what? Always revise your previous question. Even for NEET, my idea is simple. If you go through the notes, you can find some question are tricky. Like even I discussed this question that after birth there is what? Formation of this and that cause jaundice. I discuss everything. Why HbF is better? It has no what chains? HbF has no beta chain, so it doesn't bind to BPG, so therefore it has high affinity for oxygen. So we've done that already. Okay, so all these things you can go through the notes and you'll be able to solve bulk of the question. Some question might be a bit tricky or might not be covered properly, but eventually if you keep doing question, you'll be able to solve most of them. Okay, so thank you very much. Okay, I'll be finishing the class here. Okay, so hope to have more session interactions to cover bulk of this. Okay, so thank you very much. Okay. हाँ वो कर देना बंद ठीक है नहीं मैंने नहीं किया, कर दो।